Hi everybody, today I'm gonna talk about biological process which is somewhat adapted here in the Bioshock game in the storyline. And
You will have to fight me for her!
okay, so as you can see, this is a splicer, and as we will see in this sequence,
Too spliced up. I'm too spliced up. Nobody's gonna want me.
yeah, so you see in this this flashback, the lady says, I'm too spliced up. Now in the game, the splicing is probably referring to a sort of rearrangement in the DNA of these people, which used too much Adam and too much plasmids to acquire the abilities that you also have, like shooting electricity. But actually, there is a real-world biology phenomenon which is called splicing and also alternative splicing. Now, what is this? I will start with the premise. In the human, there are around 21,000 to 25,000 protein encoding genes, and in the mouse instead there are also around the same number, and also it's a similar number in certain types of worms. Now it's evident that the biological complexity of these organisms is very different, although they have all the same number of genes. So how is this happening? The thing is that we as humans can produce many more proteins from the same number of genes compared to this other species. This is not the only explanation for the increased complexity, but this is one of the explanations. And from the same genes we can produce a wider array of different proteins. I will, in order to explain this, I will use an analogy. It's a little bit like video editing. Many of you are familiar with video editing. Many YouTubers use video editing. I personally don't, because I like to keep it in a sort of conversation and do everything in just one take, for now at least. But many do, and also in movie making in Hollywood or whatever, they use video editing. So the director, for instance, doesn't have to shoot the sequence of a movie in the chronological order that appears in the final movie. In that case, basically, some sequences that are shot are then either deleted in the end product because they don't fit well, or they have to just cut it because of time constraints and how long the movie should be, and for other reasons. And also the sequences can be moved around, and also to have a different meaning of certain events, so this is actually something pretty similar as a concept of what happens inside our cells. Basically, once a gene has been transcribed into messenger RNA, this is first pre-mRNA, so it's not a mature mRNA, but it needs to be still processed. One of these processes is this alternative splicing, whereby some enzymes are able to cut this pre-mRNA, and this pre-mRNA is constituted by several sequences, which can be, in some cases, deleted from the final mRNA, or they can just be rearranged in a different way. And this deletion or rearrangement upon the translation by the ribosome and the production of the actual protein, this means that proteins will have very different amino acid sequences, or rather they will have different segments of amino acid sequences which will be arranged in different ways, and this will then influence how the protein folds, and in the end will influence its function. So this way you can really produce very different proteins starting from the same gene through this phenomenon of alternative splicing, much like in the production of a movie you would move the sequences that you shot around in order to give a different meaning or cut the movie shorter. Okay, I hope this was interesting for you, and you can hit the like button if you liked it, or the dislike button, and then you can also leave a comment if you like, and subscribe for more of these videos. Thank you for watching. Bye.